presentation, I will continue a cycle of lectures which started before summer, and it will be about mathematical modeling of respiratory viral infections. And today I will talk about one of the very serious and life-threatening consequence or possible consequence of such infection related to airway obstruction. So what it is about? First of all, of course, about human lungs, which you can see schematic representation here. And human lungs contains 23 generations of airways, starting with the largest ones and to the smallest ones. We will consider, in fact, what is happening in first 16 generations of bronchi and bronchioles, where in fact mucus is produced, and I will explain all that. Here at the left, you see a CT image, computer tomography image, with these white areas indicated by arrows, which show the area in the lung where some airways are obstructed, and of course this causes serious difficulties for respiration. And such phenomena occur in fact for many different airway diseases, and one of the most common one is called chronic obstructive pulmonary disease, COPD, but also for asthma. Another very serious genetic disease is mucoviscidosis, but also for different viral infections like influenza, and of course for COVID. We know that many people had very serious, very severe lung damage with different possible consequences, and one of them is airway obstruction and just difficulties of respiration. And here at the right image, you can see again this schematic explanation, schematic representation of what is happening. In fact, it is a cross-section of a small airway, of a small bronchiole, where you see this airway in the middle, and this yellow thing is mucus. Mucus is produced everywhere in the first 16 generations of lung airways, but if it moves, I will explain it later in more detail, and normally you see this red or violet cells and the small like black things called cilia. Cilia, I will translate, реснички, cilia. They move, the cilia move, and they put this yellow fluid, mucus, in this case towards the right. And this is very important. The role of mucus is very important. It is also very important that it moves in a proper way. But if it doesn't move, and if its production is larger than it should be, then this mucus, this fluid, accumulates in the airways, in the bronchiole, and it can completely block air circulation. So I think that you understand now how important this, how dangerous the situation can be. So the purpose of my today's talk is to discuss how we can model this situation. Please, I would like also to say, if something is not clear, I am trying to speak very slowly, very clearly, both for the language and also for the all these biological questions, later modeling questions. So please, if something is not clear, you can ask questions. Since I don't see the screen, so maybe Anastasia or somebody else, you can just interrupt me by voice and ask a question.
Okay.
So yes.
Okay, there is no questions now. For now.
No questions. Okay. Okay. So what I will talk about today is shown here. We will start with what we already know. We discussed in previous seminars and also recently about infections spreading in cell culture and tissue. I will shortly repeat what we know about this. Then I will explain and introduce this mucus motion, short biological introduction. And then we will consider this mucus motion, well, two different models. I will explain it later. So we will start with this model. We discussed it many times before, and even today in this student seminar. So I will recall it very shortly because we will use it below. So there are three equations here for uninfected cells, infected cells, and virus. If we do, for example, numerical simulations for this model, we see that infection spreads in cell culture or in cell tissue like a wave, as shown here. This blue curve is uninfected cells, green curves infected cells, brown curve is virus concentration. All that moves here from the left to the right with certain speed, c. And this is very important, and this is why all the time, or many times, we discuss this wave speed. Why it is so important? Because wave speed, the speed with which this viral infection spreads, it is proportional, it is related to virus virulence. I will, of course, we discussed it before. I will recall it with this example, which you show here. You see this two Petri dishes with this blue area. These are cells. Then they introduce virus here. This virus spreads, infects cells. After some time, the cells die and they form these white areas. So this is called virus plaque assay. So the size of these plaques, it is accepted in the biological literature, the sizes of these plaques are proportional to virus virulence. Virulence is can be interpreted as how severe then the disease is. So for example, here it is compared Delta variant and Omicron variant of the SARS-CoV-2 infection. We see that these plaques are much larger for Delta than Omicron, so Delta virus is more virulent. Of course, there are many details. We discussed it previously. We will not discuss it now. Also, we see here this image showing the lung, and these yellow areas are the areas of the lung damaged by this COVID disease. So roughly speaking, of course, it's very approximate. So these areas damaged by virus are determined by virus virulence. But the sizes from the mathematical modeling, we know that the size of these plaques depends on the wave speed. In fact, this is the speed with which these white areas flux grow. So everything become related. So we determine in mathematical model the wave speed. Sorry, that's a grammatical mistake here. And this wave speed determines virus virulence. Another important characterization is viral load. Also, we can determine viral load in the mathematical model. And why it is important? Because viral load in the upper respiratory tract, you see here, determines the infectivity of this virus, how fast this infection is transmitted from the infected to uninfected individuals. So these two most important characterizations of viral infection, virus virulence, and virus infectivity, we determine in the mathematical model. Then we apply these results. We study different variants of, for example, this SARS-CoV virus, like Delta and Omicron, for example. We compare with experimental results. We determine all these characterizations, speed, viral load, and so on. We discussed it several times. I will not stop in detail today. So this is in some sense for us now, it's some kind of a basic model and basic results, meaning comparison with experiments, biological interpretations. Then we have done, on the basis of this model, we have done several developments, in particular here, virus competition model. There's now two viruses. They compete with each other for uninfected cells, and then we do some analysis. We compare with experimental results, and our conclusions corresponds to the experiments and to clinical observations. Another development of these models is related to virus mutation. Also, there are two viruses here in the model, and they can mutate one into another. Also, we studied this model. Another development is shown here. We started with 3 equation models. There are 4 equations now. So from uninfected cells to infected cells, we go now not directly, but through exposed cells. Exposed cells, these are cells which are already infected by virus, but which do not yet produce new viral particles. Well, another interesting development, we introduce the immune response in the model with different immune cells and interferon, immune cells, antibodies. One recent work in progress, we study intracellular regulation of virus replication. You see, there are no more infected cells in the model, but we consider this intracellular virus. So there's extracellular virus. It enters the cell as intracellular virus. Then there is viral RNA here. There are cell proteins A. They produce new viral particles which go outside the cell, and so on. So you see, we have done a cycle of works devoted to viral infections. These works are continued with several PhD students now involved in the continuation of these works. In particular, we study infection with inflammation, multi-scale models of infection, and different inflammatory diseases. So this was an introduction about mathematical modeling of viral infections, like a short overview of recent works we have done in this area. So, and now I will talk more specifically about viral infections in the human body, in the respiratory tract, and about mucus flow. So what it is about? Here you see at the left image, you see epithelial cells. So you can think about human lung, airways, bronchiole. So the first layer of cells, so here, this white area, this is air coming into the lung. Then there is this, they call it epithelial cells. Epithelial cells. That in this epithelial layer, there are different cell types. In particular, you see here shown goblet cells. These goblet cells, they produce mucus. Mucus, well, better to say, to be more precise, I will explain it later. It is what is called airway surface liquid. I will explain it a little bit later. Anyway, these goblet cells, they produce mucus. You see this like layer here. And then there are also other cell types they call ciliated cells. You see this cells with this cilia here, реснички по-русски, cilia. And it's very amazing, in fact, how all this organized. It's not very well understood. It's very complex. You see now on the right image this cilia, and in fact the cilia move. They move in a periodic way, in a very organized, very coordinated manner. They move all together from one side to another, and they push mucus. So, summarizing this is important to understand. It is a very important physiological process. So there are epithelial cells, there is mucus, there are cilia, and the cilia push mucus. And this mucus moves along the lungs from the bottom upwards. This is shown here. You see in the left, it's schematic representation of the lungs with these consecutive generations. And these airways, they become smaller and smaller. Then in the middle image, what is shown, it's all airways in the same generation, they put together like rectangles. So we have like this rectangular domains, and the total width for each next generation becomes larger. You see, somehow, oops, interesting. Somehow I have a pointer then disappeared. I don't know how it happened. Let me see. Okay. Anyway, looks like what presenter yes, pointer. Yes, okay. Okay, so this is important to understand. This bronchiole becomes smaller, but the number increases. So the total size, the total width of this of each next generation becomes larger and larger. Now let us look at the right figure here. Again, it's a schematic representation of this epithelial layer, which is below. Then there are two different liquids which form altogether. They are called airway surface liquid. You see this vertical lines. This is cilia. So the liquid which is inside below, it is called periciliary liquid. It's like water. It's not viscous, just like water. Mucus, which is above, it is not like water. It is quite viscous. In between, there is a extraction layer. It's already mucus. So the cilia, they're basically inside this periciliary liquid, low viscous liquid. But they a little bit, they enter mucus. So when this cilia move, they push mucus because they're here a little bit inside mucus. This is how it happens. Before we started this mucus flow to model mucoviscidosis, as I mentioned, it's a very serious genetic disease. Now we will apply this model for viral infections. So let us begin with the first model. I call it local mucus motion. What this means? We just consider a small portion, not the whole lung, but a small portion just of one bronchiole. And then we have here, we consider two layers. The first layer, the lower layer shown here in this figure, these are cells, epithelial cells. These red ones, these are infected cells. So this infection propagates here from the right to the left. It's shown with this blue arrow here. At the top of these cells, there is this airway surface liquid, which moves here in the opposite direction. So we write mathematical model. These three equations in the left, you can recognize the same equations for uninfected cells, infected cells, virus. Now, what means virus here? You see it's written VC. It's virus in this in the cell layer, in the cells. And then there is also exchange of, so this virus from the cells, it goes to the liquid layer. So there is also this V, it's virus concentration in the liquid layer. So there is an important difference between these two equations. Let us consider this equation for VC, virus in the cell layer. What is important here? That virus is produced in the infected cells, this term. But there is no production term in the second equation because virus is not produced in this liquid. Then there is exchange between in the virus between cell layer and fluid layer. I will not discuss that. Then if we consider this virus in the fluid layer, what is important here also is that convection, so this airway surface liquid, it moves with certain velocity. So this term is shown here. So virus in the liquid, it moves together with this liquid. This is very important. In both equations, there are diffusion terms. So we analyze this model. I will not stop on that, and I will give you only one important conclusion. Okay, no, some modeling is shown here. Okay, there are some figures. In fact, the original model is two-dimensional. We reduce it to one-dimensional model with two layers for cells and for liquid, virus in the cell layer, virus in the fluid layer. I skip all these details and come directly to the conclusion. And the conclusion is here. So we have this cell layer, infected cells shown in red, and there is virus diffusion in the cell layer. Then from the cell layer, virus also goes to the liquid layer, and it also diffuses here. So the question is whether both of these diffusion processes are important. So if we suppose that there is no diffusion in the cell layer, or if this diffusion, virus diffusion, I mean, is very slow here, and the diffusion occurs only in the liquid layer, then what happens? If diffusion occurs, so I mean, virus should be transported from this red region to this white region. We know that virus is produced in the infected cells, then it goes out of the infected cells here, and then because this flow velocity is relatively high, virus cannot move to the left. It is removed by convection. It is removed by flow velocity outside. So important conclusion is that in fact, virus diffusion, of course, it occurs at the same time in the cell layer and in the fluid layer. But what is important that it is basically occurs in the cell layer. In the cell layer, convection, fluid motion is not important. This is why virus can propagate along the cell layer from the right to the left. It cannot propagate in the liquid layer because it is removed by fluid flow. So this is important conclusion of this work. In fact, it shows with mathematical modeling that virus diffusion occurs in the cell layer, and that since it occurs in the cell layer, then in fact it appears that in fact fluid flow is not important for infection propagation in the epithelial cells. This is a strange conclusion, in fact, because what it means, we study the influence of fluid flow on infection propagation, and we conclude that it is not important. So I stop on that. If there are questions, please go ahead, because we just came to one important point in this presentation, this conclusion that when infection propagates in human airways, fluid flow, mucus flow, in fact, has a big influence on this infection. This is a very important conclusion. Why? Because when you read all this literature, it says that fluid flow, mucus flow plays a very important role. Yes, indeed, it plays an important role, but in a different sense. In particular, mucus, which is here, it protects epithelial cells from virus, which comes here, from bacteria, which comes here from the air. But once virus penetrates already epithelial cells, then it propagates along the cell layer, and fluid flow is not so important anymore. This is we should understand, and we use it in all this modeling. Okay, if no questions, so we continue now with the model which I call global model. We take now into account all 16 generations of human lungs. But before continuing with modeling, I return to the biological questions, and we discuss now two important questions. In fact, how viral infection influences mucus production and mucus properties. And the conclusion from all this biological literature, and especially there are many works last 2 years because of this COVID-19 epidemics, there are two main conclusions. The first main conclusion is that because of viral infection, mucus becomes much more viscous. And the second conclusion is that its production increases. This is called hypersecretion, increased secretion of mucus. So let us discuss a little bit these two questions. This schematic representation, which I took from this paper shown here, it explains, I think, in a good way what happens, in fact. Let us see. Okay, the small viral particles shown here at the bottom left, what they do? First of all, they penetrate epithelial cells, they infect them, and then they infect also other cell types, type called mast cells. These mast cells, they produce, we discussed it today and we'll discuss it before, this what is called inflammatory cytokines. So they initiate inflammation. In particular, these two very important molecules called TNF. TNF is tumor necrosis factor alpha, and interleukin-1 beta. So these infected mast cells produce these inflammatory cytokines. They start inflammation. So you see this, then it goes this way, in both ways, in fact. Infected cells produce, infected epithelial cells also produce inflammatory cytokines, but inflammatory cytokines also act on cells, and both together, this inflammation and infection itself, what they do, they change the composition of mucus. In fact, mucus contains many different molecules called glycoproteins of different types. In particular, there are these mucin molecules, it's called mucin molecules MUC5AC and MUC5B, and because of inflammation and because of infection, the concentration of these two types of molecules increases. And in fact, biological literature says that they correlate with this mucus viscosity. More molecules of this type, more the mucus becomes more viscous. It is written sticky here. Okay. And you see this at the left, it is written intubation, ventilation. It means that because of this sticky mucus, it is not properly removed. So there are this mucus accumulation in the airways. Because of that, it is necessary to do intubation and lung ventilation. So this is a short schematic explanation. Of course, there are many other phenomena, but this is what is most important. Here it is basically the same, but from another paper, another schematic representation. If we look at this right image here, we see at the left normal case, at the right the SARS-CoV-2 infection. You see this yellow layer, it's mucus, it is called mucociliary transport. It's a narrow layer of this mucus. At the right, when it is infected, this layer becomes very thick. What we should also know that mucus produced not only in the goblet cells shown here, but here it is called this part, it is called submucosal gland, also mucus produced here. And if we look at the left, what is shown here, SARS-CoV-2, airway inflammation, inflammatory mediators, mucin overproduction, and so on. Look, mucus plugging, severe pneumonia and lung infection, death. This is what often happened during this epidemic. This complex scheme shows how virus acts inside infected cells. There is this whole chain of events, and finally, at the end of this long chain, we have this yes, here you see this mucus hypersecretion and so on. Yeah, here this mucus. Okay. It was for virus, and the next one is for this inflammatory cytokines. Also, it's explain how this inflammatory cytokines act, and all that finally also act on increased production of mucus. Okay, now I will show you the model which we use to model all that. So, in fact, it contains two submodels. The first submodel is almost the same as before. So, it is for virus. As before, we have this uninfected cells, infected cells, two-layer model, virus in the cell layer, virus in the fluid layer. The only difference here is that we now introduce this H. H is the thickness of the fluid layer. I will not explain in detail these equations, but it's not very difficult to write them. And the second submodel, it concerns mucus motion. So it includes, in fact, three variables. So H is the thickness of this layer, fluid layer, and this is equation for this H. And in fact, so in the right-hand side, there are two terms. The last term, it shows mucus production here in the epithelial layer. And the first term, it shows this convection, fluid flow, and this fluid flow changes this also H, the thickness of the layer. In fact, it's a simple equation, and it can be obtained from the conservation of mass. It's only conservation of mass written in this equation. It's not a long derivation, but I will not stop on that. So we have conservation of mass, and this equation gives us this thickness of this layer H. Another equation for the viscosity, because let us look at this last term in the right-hand side. You see here, it's uninfected cells. They produce mucus with some viscosity called mu0. It's a normal viscosity. Then this I infected cells, they produce mucus with high viscosity. So it's mucus production here by uninfected cells and by infected cells. Then this first term, again, it's convection, motion, this of fluid flow, it's we should take into account because it moves this fluid with different viscosities, so it changes also viscosity due to convection. And finally, this diffusion of these mucin molecules, it also changes viscosity. So again, it's a, well, it may seem a little bit complex, but in fact, it is a simple equation just for the conservation equation, like a conservation law with diffusion. The last equation is for fluid velocity. You see, in all these equations, we have these convective terms. So again, it can be derived, but to make it simple, it is observed that this fluid flow depends on viscosity. And how it depends on viscosity? Mu here is the viscosity of at some places it's this variable viscosity found from this equation. Mu1, it's viscosity produced, it's a constant, it's viscosity produced by infected cells, and it's a very high viscosity. So when mu approaches this mu1, this some very viscous mucus, very high viscosity, you see. So if mu approaches mu1, then flow velocity goes to 0. This is a quite simple approximation, but this is what observed experimented. But this is very important for us. You will see right now that when this viscosity increases to this maximal layer, then fluid velocity goes to 0. Let us keep it in mind. Okay, then, well, some simulations. This is an example of simulations without infection. We just impose some value of viscosity here, different values, and we obtain distribution of this H thickness of the layer. Why it is like steps like that here? Because each step corresponds to one generation of airways. And for example, if you take the first one and the second one, I told that in the beginning that there are more and more of these airways in the next each generation. So mucus moves here from the right to the left. When it goes from the second generation to the first generation, so the width of the layer becomes smaller, so H increases. Hopefully it is clear, since I don't see anybody. What?
May I ask a question?
Yes, please.
I thought H is the width of the mucous layer.
Right.
You said it is inversely proportional to it.
To inversely, no. H is the thickness of this. No, there are two parameters. Okay, let me maybe return to here. You see, there are two things. The width, it's like, you see here, this is the width.
Mm-hmm.
Anastasia, I think I recognize your voice.
Yes.
So you see, each next generation, there are more and more airways in each next generation. Total width becomes larger. Is it clear? If you compare the first generation, only one rectangle. In the second generation, there are two rectangles. Each rectangle, its width is smaller, but the sum is larger. Consider now mucus, which moves from the second generation to the first generation. Since the total width becomes smaller here, the thickness H becomes larger. Is it clear?
H becomes larger in the generation with smaller number.
Yes.
Mm-hmm.
Yeah.
Yes, I think I understand.
No, I really want you to understand that because it's simple. Like, think about the river. So, so the river somewhere in the plain, it is very wide. Then it goes to some mountain place, it becomes narrow, right? In the place where it becomes narrow, the water level becomes higher. Is it clear?
Yes, yes.
So it's exactly that. So our river becomes narrow here, so the level of the water increases.
Yes,
because it moves from the bottom up, it becomes more and more narrow, and so more and more deep. So this is what is shown here. You see,
so the first generation is
the first generation is the largest.
Zero.
It's the largest, yes, here. Then we go to the right, and here it's the total length. It's about 24 centimeters. Of course, it depends on the person, but like an average, 24 centimeters. Here, there are very, very small airways, very, very small bronchioli. But there are many of them, many thousands, tens of thousands of them. So yes.
One more question. These levels, for example, for the second level, it is the thickness of mucus layer in one airway.
It is the same.
Airways.
No, it is the same. It is the same in all airways of the same generation. If you take,
one, not some of this.
No. If you take, for example, here the first one is only one here. Then there are two of them here, and the depth, the thickness is less here. And then we go right to the last 16th generation. There are 30,000 of them, but they are very small. H is very small here.
Yes, thank you.
Okay. Then we take different values of viscosity from normal viscosity at the bottom to very high viscosity. So we used this maximal value of viscosity. We considered here it's 0.16. For 0.16, it's the maximal viscosity. The fluid doesn't move. Let us take 0.15. The difference is small. So this H, it's this upper curve, right? You see that? Then we compare this H with the diameter of the airway. There are 16 generations. For each generation, we compare H with the diameter of the corresponding bronchioli. Anastasia, since you are here and looks like you listen to me, tell me whether you understand what I mean.
Yes, I understand, and it was my next question.
For example, we take this left part, the first generation. So it's given here in meters. So here it's like, let us think, if we take it in centimeters, well, it is like it's 0.3 millimeters. Okay, 0.3. It's less than this. H here is less than 1 millimeter. Okay, but the diameter of this airway is 2 centimeters. So even if we take the maximal viscosity here, well, not maximal, but very close to maximal viscosity, H is much, and it is the same for all airways, even more for small airways. H is much smaller than the diameter. There is no any obstruction. There is no difficulty with respiration. It's not a problem, even if we take such a large viscosity. So it's not like that it works. So let us see. Okay, this is again all these equations. We discuss them. Okay, now expecting a little bit where I'm going to come. In fact, there are, from the point of view of mucus and lung obstruction, there are 3 stages of infection progression. So look, let us consider this first stage. In fact, if this red line shows how infection, it is a concentration of infected cells, red line. Changes, so it's adimensional, changes from 0 to 1. So at the left here, infected cells, at the right, uninfected cells. This curve moves from the left to the right because infection propagates like that. Here, what I show corresponds to the situation where first 4 generations are infected. First 4 generations are infected. Red line here. This is H, the distribution of mucus. This is of thickness. Mu, distribution of viscosity. V, distribution of velocity. To make it short, practically nothing happens when infection comes. You see, with generations are very different. Big generations, small generations. This first 4 generations is already more than a half of the lung. More than a half of the lung of bronchioli are infected. Absolutely nothing happens because because H almost exactly as before. If you look at mucus viscosity, you see the values. It was 48, it was the minimal. Now it changes between 48 and 61, so a little bit more, but it's absolutely inessential. Nothing changes. 4 generations. Now next, we take 10 generations infected. You see a red line. It's almost at the end. It already 90% of bronchioli is infected. If you take the length, it's 90%. But according to generations, it corresponds to about 10 generations, which are infected. It's here. And we do the same. We take H, we take mu. Okay, mu changes. Look, so, 48 to 84. Okay, changes more or less twice. But this change again is very small. Nothing happens. If you take, I took for example, as example, this generation number 10, and we measure H for the generation number 10, and we obtain, you see this many zeros and then 3 in centimeters. And if we take the same generation, not 10, maybe 9, here, and look at the diameter, it's written here in the table, it's 10,000 times more. It's absolutely no problem. The width of this layer is thousands of times less than the diameter of the bronchus. You see, it's amazing, in fact, that this almost everything is already infected, but from the point of view of mucus accumulation, nothing happens. So this is what I interpret as the first stage. So infection, well, I took this 10 generations, but even more, if we continue with 12 generations, it's practically no influence. So the conclusion about this first stage of infection progression, we take 12 generation. It's more than 90% of length of the bronchi. Small change of viscosity, small change of layer thickness, and it remains thousands of times less than diameter of the corresponding bronchi generation. So nothing happens. So then second stage, it's when this infection continues from the generation 12 to generation 16. In fact, it completely infects all this bronchioli, all this part where mucus is produced. Well, in the model, it takes about 7 days. In fact, the number of days is not important because our infection propagates with a constant speed. I think that in this case, propagation is a little bit too fast. Maybe in reality, it takes maybe not 7 days, maybe 2 weeks or something like that. So what happens then? In fact, it's a very short stage, quite short, because if here it was like between 6 and 7 days, then next several hours, maybe 10 hours more, and then this infection completely propagates along all these generations. And what happens? Everything changes now. In what sense? Since all of them are now infected, all of them begin to produce viscous mucus. This is what is important. Why, in fact, I return to this first stage. Why nothing important happens here? Because the most part of mucus is produced here in small generations. And here, mucus is normal. You see, everything is infected except for this small part. But the most part of mucus is produced exactly in this small part. And mucus there is normal. This is why nothing serious happens till now. But if we return to this case, so everything now is infected. And viscous mucus is produced everywhere, everywhere. You see this low figure? So this mucus viscosity here at the end is 0.157. I told you that the maximal we consider 1.6, and now it's 1.57. It's already close to the maximum. And because of that, mucus velocity drops down. It decreases strongly. It decreases already at this stage about 10 times. So at the end of the second stage, mucus viscosity becomes quite high. Mucus velocity drops very strongly, like roughly 10 times. What happens with age, with the thickness? Thickness is still very small. Maybe not 1,000, but maybe hundreds of times less than the diameter. So no problem for thickness. You see, the second stage, what happened? There is a problem with mucus viscosity already. There is a problem with mucus velocity, but not yet with the thickness of this, because just thickness grows very slowly, but this change was very rapid. So thickness remains small. This is the second stage. So what we conclude here for the second stage, flow velocity from 10 to 100 times. And in fact, this plaque is formed already now. Mucus viscosity approaches maximal values. Layer thickness remains very small compared to the diameter. And now the third stage. What happens at the third stage? So flow velocity is very small and converges. You continue to converge to zero. Mucus viscosity continues to grow and converges to the maximal value. And now we take our model and we substitute in our model flow velocity 0. It is not yet exactly 0, but it is very small and it converges to 0. So let us just do this exercise. We substitute v equals 0 in our model for h, and we obtain this very simple equation. Very simple equation, just linear equation. Just j of x is how fast mucus is produced depending on the position. Okay, I will. Here it does not depend on time. I will comment on that later. So dh dt equals this function. It does not depend. So for each x, h grows linearly as a function of time. This is our important conclusion. So we take any point in our x, any point x in our this bronchial tree. We take this position, and at this point, h is a linear function of time. H is small. We say that in the previous stage, h is small, but it continues to grow and it grows linearly, and this is important because after several days more in our simulations, it corresponds to before it was 7 days, so several 5-6 days more. Because of this linear growth, the thickness of the layer grows linearly in time, and it becomes already comparable with the diameter. Here, after 12 days, we take this generation number 15, 14, 15 here. So the thickness h becomes already 0.03 centimeters. And here the diameter of this generation is 0.07, taking into account that it happens from both sides. We multiply h by 2 and we arrive almost to that. So the conclusion is that we a little bit later, now the airway is very, very narrow. A little bit later, we obtain complete lung obstruction in this small bronchiole, and this is exactly what is observed for this, in particular, COVID patients. For large airways, it's not so dramatic. This obstruction begins in these small airways. So this is basically what I wanted to explain. And the conclusion is that infection wave passes through the bronchial tree. Mucus viscosity increases in infected tissue. Mucus flow stops. Layer thickness grows. And all that leads to this airway obstruction. And this process, as we discussed, occurs in 3 stages. The first very long stage, when almost all bronchial tree is infected, practically nothing happens. Then very rapidly, last generations, when last generations are infected, mucus viscosity drastically change, mucus velocity drops to 0, and then the third stage, nothing moves, the plaque is formed, mucus thickness increases linearly in time. Several days after that, it's finished, complete obstruction, the patient dies. Okay, this is what I wanted to explain. There are a number of comments, remarks. We don't have time for that now. Also about treatment in particular, I give here an example of treatment. If we use the same treatment as is used, for example, for mucoviscidosis, it decreases this treatment. It's like an aerosol inhaled. It decreases mucus viscosity. The problem is that these droplets, they do not penetrate to this last generation. So this viscosity is decreased here, but not here. And in fact, most likely this treatment will not give anything. Okay, I stop on that. Let's me. Okay. Okay, please, if there are questions, so please you can ask them now.
And did you do the simulation with treatment? You mentioned, but not.
Yeah, we did simulations. Well, I showed you an example of these simulations. In fact, we started this treatment in detail when we started. Well, it was like years ago. This mucoviscidosis. Mucoviscidosis is a disease, a genetic disease where mucus is more viscous than it should be. It's a very dangerous disease, and usually people don't live long with such disease. And well, there are some treatments which, like as I mentioned, you inhale special products, they decrease the viscosity.
And is there from the model any suggestions what the treatment would be effective? And you said,
well, I think that in this case, modeling cannot really suggest the solution of this problem. What they do clinically, they do intubation. It means there is a tube inserted in the lung. It sucks mucus. You understand what I mean?
Sucks, not what is
well, высасывает mucus. Понятно? Well, this is the only solution, the only efficient solution, but still it is, well, it improves a little bit the patient condition, but what is of course very important is to stop infection and inflammation. If it is not stopped, then I'm not sure that, well, it can really solve the problem.
What's the reason of mucus flow? It is the cilia cells, yes, they are moving this mucus. May we somehow affect the cells so they can?
No, truly speaking, I don't. I don't think that this is possible. I don't know. I cannot say. I have not heard about such things. So in fact, it's very impressive. You can probably find like videos or something like this cilia. They oscillate like that all together. Like they do this motion like that, and this is really a very, very complex, very interesting question for modeling, because they move like that, then they return to their position, they move again like that. So they move both ways. Why they push it in one side? You understand the question?
Yes.
The cilia, they move from the left to the right, then they return from the right to the left, but they push. No, this is exactly the point. Not symmetrically. Their motion this way and this way is not the same. This is why they push mucus only in one direction. So such amazing things. It's very complex, but it works. Normally it works.
Yes. Thank you. I have no questions anymore.
Okay, I think that our audience doesn't have other questions. So
maybe Joseph.
Yeah, there is what? Yeah, please go ahead.
if the secretion of mucus 5b and the mucus increase by the cilia cells, we have a more viscosity.
Repeat, I don't understand.
If we have more secretion of mucus 5b and the mucus
AC, yeah.
we have more viscosity.
Yeah, mucus is more viscous. Yes, because mucus is composed from many different, they call it mucin. These molecules, glycoproteins, they call mucin. There are many different types of this mucin, and according to the data, biological data, more viscous mucus correlates with the concentration of this 5B and 5AC, right? And because of infection and because of inflammation, the concentration of these two types of mucin molecules increases. This is why mucus becomes more viscous. I did not discuss in my presentation today this hypersecretion. It's not only it is more viscous, but there are also more mucus produced.
Yes.
From my point of view, what is most important is viscosity, because we saw, well, what I showed you in the model. So mucus layer, it's thousands of times less than the diameter. Okay, we increase its production by 2. Okay, it will be not thousands of times, it will be 500 of times less than. It doesn't change too much. It's not that. It's because of its viscosity increases, its velocity drops to 0. This is what is important. Okay. So please, I saw other hands, but they disappeared. So please, if other questions, please ask. If not, we'll. 9. Who is that? Please, there are some. I don't understand with this raised hands. Please, if there are questions, then please ask. If no, then we'll finish our seminar. No, it looks like okay.
Maria had raised hands, but she, I don't know. Do you have questions, Maria?
I have a question, but I can't talk now. I'm sorry.
Maybe I think you can...
Вы можете спросить по-русски, если вы не хотите, или не можете спросить по-английски.
Я могу написать.
Ну напишите.
And while Maria asked a question, may I ask one more question? You said that there is a pre-cilia liquid and mucus, and they are connect to each other. One layer and another. Can they mix and
No. I don't know exact answer. Well, my hypothesis is that first of all, what is for sure that they are separated. Maybe at the interface, maybe there is a little bit of mixing. What I started to say is that I don't know whether these fluids are miscible or immiscible. Okay. Смешиваемые и не смешиваемые. It's a very interesting question. We studied it for years in completely different contexts, miscible and immiscible fluids, but I will not discuss it now. I think that they are I think that they are miscible, but not highly miscible. So there is a small, narrow transition zone between them.
So it cannot, the pre-cilia liquid cannot influence the mucus.
Mucus?
Yes.
Well, in fact, of course they are connected because, of course, it's much more complex than I presented. Because this periciliary liquid, it's not viscous, it's like water, but it is important to have it's this layer sufficiently large as it is supposed to be, because for in some other situations this layer becomes also narrow, and this cilia becomes a bigger part of cilia enters mucus, and since mucus is very viscous, they cannot move. Cilia cannot move anymore. You understand? Cilia move normally because their most part are inside this periciliary fluid, low viscous. This is why they do this motion. But if they are completely in mucus, they just cannot move. And again, it stops. So in fact, everything is connected, many other things. I'm not really specialist at these questions. Yeah, so in chat, what is written here? Why are cilia moving? Are they moving by air? Certainly, they are not moved by air, because in fact, they even don't touch air because they are completely inside fluid. Yes. So why they move? Truly speaking, I don't remember whether before I read about the mechanism of their motion, I cannot answer this question. So I think that these are cells. So cilia by themselves, they are completely inert. There are no mechanisms. There are no motor which move cilia. It's kind of, it's like hair. Hair, you see, they cannot move themselves. Of course, the structure of cilia is more complex than hair, but like to explain. So they cannot move by themselves. It's cells somehow produce this motion, right? But how it works precisely, I don't know. I cannot say. Okay, no, well, Joseph, I don't know, your hand is raised. You have another question, or you just?
No,
it's
just,
okay. Yeah, okay. No other questions. Then I would like to thank all our listeners and also our speaker. And we stop for today. The next time, next week we have, well, I think we have a talk about blood coagulation. I forgot the name of the speaker, but Tatiana Kovalenko.
Different modern approaches in the simulation, yes, coagulation factor 10 activation.
Yeah, but Anastasia, how it will be done in Russian or in English? What?
I think we can ask speaker if she can present in English.
Yeah, please do that. Ask her whether she can do it in English, right? Because we have our listeners, not necessarily Russian speakers. So please ask her. If occasionally this is not possible, then in both cases we will send this information to confirm about the language.
Okay, I ask.
Okay, thanks to everybody and have a good day. Bye.
Thanks. Thank you.
Спасибо, Виталий Айзикович. Спасибо.
Спасибо.
Thank you very much.
По-моему, это был Марк Балабут, да? Марк,
Так точно.
Марк, да. Приятно услышать вас голос.
Спасибо. Я
Спасибо.
что-то понял. Да.
Спасибо.
Да. Ладно, всего хорошего всем. До свидания.
Thank you.